Good morning. Good morning, everyone. We will be starting the workshop in another 5 minutes. Good morning, everyone, and a very warm welcome to the workshop on understanding forest phenology using phenocams. So on behalf of the organizing committee, we extend our appreciation to all your presence today. So let me begin this day by thanking all the wonderful teachers who have guided us, inspired us, and given the knowledge that lasts lifelong. So our sincere gratitude to all our teachers today. My name is Shruti, and I'm the project scientist from the Department for Climate Change at EMPRI, and I'm also the facilitator for today's event. So it's with immense pleasure that I welcome our distinguished guests today.
Recording in progress.
So their presence here adds immense value to this one-day workshop and promises to enrich our understanding in the phenology monitoring using Phenocam technology. So before we proceed with the training, I would request Dr. Jagmohan Sharma, the Director General of EMPRI, to set the tone for the workshop. Dr. Sharma is the member of Indian Forestry Service serving on Karnataka cadre. He holds a PhD in Climate Change and Forests from Indian Institute of Science, Master degree in Public Policy and Management Study from IIM Bangalore, Masters of Science in Forestry, Ecology and Management from University of Edinburgh, and Masters in MTech in Rubber Technology from IIT Kharagpur and MSc in Chemistry from IIT Kanpur. He's published several papers in both national and international journals, focusing mainly on vulnerability and risk assessment under climate change, carbon stocks and flux, forestry, wildlife ecology, and water yield from various forests. Dr. Sharma has held various positions in Government of India and Government of Karnataka in forestry, wildlife, environment, and water resources sector during the past 3 decades. He's presently working as the Director General EMPRI at Bangalore. So we'd like to.
Thank you very much, Dr. Shruti. I think we better I be taken off the screen and I would do that. So we'll stop sharing the screen. That's it. Yeah, thank you so much. Good morning, everyone. The esteemed guests and very important resource persons for today's webinar. Dr. C.P. Singh from the Space Application Center, Ahmedabad, Dr. Rajiv Chaturvedi from BITS Pilani campus at Goa, Dr. Suresh from Indian Institute of Science, Center for Ecological Sciences, Dr. Shruti, who is heading this project, the staff, scientific and others from Department of Climate Change, and a number of persons interested in the subject who have joined us online. I welcome all of you to this one-day webinar, which in our opinion is a very significant step in monitoring of forests in the country. Ladies and gentlemen, the state has given to itself Karnataka Action Plan on Climate Change, and therein may I please request for switching off your mics and switch them on only when you're talking. I was mentioning about the climate change action plan that the state has given to itself, wherein there are several sectors about which we have talked in terms of the vulnerability of those sectors, the risk that they have under climate change, and what state can take as adaptation measures primarily, and the mitigation opportunities that are there in those sectors and in general. In that, forestry sector is an important sector. We understand natural resources are always climate dependent, and if climate changes, it will have implications for these natural resource sectors, which includes forestry, which is the storehouse of biodiversity we understand in the terrestrial ecosystems. And in that, we have undertaken risk assessments, we have taken vulnerability assessments, and an important thing that comes out of all that discussion is about keeping the track on about the changes that occur in forests. If we change in climate, the forests are going to change or are changing already in terms of the processes that happen inside forests, say regeneration. Are forests regenerating? Are forests experiencing droughts, or is there a situation of in some areas because of change of rainfall that the regeneration of certain species is more compared to the others, and hence change in crop composition is expected to happen. So while all this is natural and we may not be able to do much about the natural element of it. However, we understand there are certain man-made factors like, say, fire or grazing and such other interventions that we can understand as to how the changes in forests that are occurring because of change in the natural parameters, how this is man-made factors are having impact. And if we can manage them in order that forest transition can happen, we are expecting that unless there are long droughts and there is a dying back of forest, that transition should be possible given we are able to manage the anthropogenic factors. And such transition, obviously, it takes time. We do not know how much time we have. We have no information about the thresholds. But we would want to monitor forests in order that we know what is happening. At least that information has to be with us. And this is people trying to do this globally. And we would not want to be left behind in this, because India hosts biodiversity in forests, and so much of endemicity is there that we cannot neglect it. What management options are there can also be evolved from what the changes we see to the capacities humans have and at the skills they can work. Nature operates at a much larger scale. We cannot match it. So in this background, this institution proposed a project to Department for Science and Technology, which had phase 1, during which some monitoring of forests was undertaken. We already have at least 3 permanent preservation plots where we are making observations. But in the second phase of DST project that we got now, we are aspiring to undertake monitoring exercise using technology. And Phenocam is one such opportunity, a tool that not only is able to capture data about the changes that are occurring, phenological changes, but also the meteorological factors as they exist when such changes occur. In order that we perhaps, if we have computer-based programs, would be able to look at it and see if there are any correlations between them and what is the pace of change that we'll be able to decipher. So under these 2 DST projects, we have high aspiration. However, the experience that we have is only of working at a ground level. We go manually to a spot and we take measurements. But how this tool will help us, what is this tool? what is its nature, what are its capabilities, how this can help us monitor, and the data we get out of it. If we can make use of that data, do we have capacity to analyze that data? What additional resources we might need in terms of the people who have capability to process such data? All we need, we are in the process of discovering. The research team is looking at the literature available in this regard, and we found that we do need hand-holding at this stage. And that is how this workshop became very important for us, and particularly for the team, in order that they are able to understand the basic requirements of the project they are undertaking, and also the people who are active in this field who can, when requested, could extend help. And I trust there are a number of people with good experience who have joined us online. They are part of this webinar, and on each other's call, we could be available for providing mutual assistance. We also understand that at national level, there is an effort for monitoring of forests, which is ongoing. It's a long-term thing. It has to be long-term, and we wish to be a part of it through this initiative. And we very sincerely think that in the Western Ghats of state, where we host most of our forest, and which is highly biodiverse area, if we are able to set up preservation plots which are monitored through these tools, it will be a useful contribution, and in a sense be adding completeness to the national project that at present is. And indeed, we want to learn from the national project about what protocol is being followed there, so that we do not have our separate protocol which does not integrate with the national protocol that is available. So those are among the important questions that we are asking ourselves, and we would try to find answers to those questions today in this webinar. We basically want to have 3 preservation plots in 3 different types, is what our initial thinking is suggesting. Unless in the process of discovery, we find that it's useful to, say, suppose within evergreen forest, do at 2 places rather than going to dry deciduous forest. But all that discovery has to happen. So the 5 important questions which we are wanting answers for through today's workshop are as to which forest type we should monitor. An answer for that perhaps would come from what is being monitored where in the country, and where is the gap. If no one else is monitoring a particular type of forest and we have that in Karnataka, then that gap we would want to fill through this project. Question number 2 is about at what location we could be establishing these permanent preservation plots. That's very important. Such a location not only has to be such that where it's possible to isolate the climatic impacts from man-made impacts, or to minimize them, in order that we can attribute the changes that occur largely to climatic parameters. The third question is about the parameters, as to what do we monitor there? On what factors we collect information in those plots. The fourth question is about what information on those parameters we collect, because Phenocam may have capacity to collect data on number of things, and we may or may not require them. And if we record all that data and aspire to use all of that, then that's challenging, though we should be doing that. But I think there would be 2 stages in which we do it. We find certain data which is very directly connected, and we could be in our capacities use, and there could be larger data that is available, and that is analyzed and used with the assistance of any other agency which is better informed and resourced to do that. And the 5th question is about what monitoring protocol we adopt, what seasons we take information, at what frequency we make observations. All those questions are important to us. And there could be certain other questions which have not occurred to us. And they would come to light as we get into the project. And that is that is one of the important factors for which we thought through this webinar, if the community that is monitoring forests in the country today has an opportunity to be on the same platform and tomorrow to cooperate, then this workshop is of a great use to us in EMPRI and everyone else in the country that is similarly monitoring the forests in the country. So I think this is a perfect occasion when we have best brains available to us in this field. They are very experienced people. They are well informed about the trends that are seen at national level and also at the global level. I also think that the information that we generate through this project will be possible to integrate with the efforts at national as well as global levels. So with this, I would welcome you once again, our resource person, Dr. C.P. Singh, Dr. Rajiv Chaturvedi, Dr. Suresh, to this workshop. And I would request them that please today assist us in finding answers to all these questions and this partnership in that sense, which starts today. I hope we'll get the benefit of your handholding of the young scientists and researchers that we have here, and you can count us as one of the centers who are undertaking this work, and which will of course be also considered at national level, because this is a DST project under which we take it. So thank you very much once again. And I'm very happy to be here today and deliberating on this subject. And I would request all our young scientists, young minds to involve themselves fully, ask those questions and try to identify answers to them. Thank you very much.
Thank you, sir. Thank you for setting the context for this workshop. I now request Mr. Balasubramanian Sharma, research scholar at DCC EMPRI, to present the phenology study that we do at EMPRI. And this will be followed by a presentation by our SRF, Mr. Kiran, who will be presenting about what we hope to accomplish in the coming days using Phenocam technology.
Good morning, distinguished members, colleagues and friends. I'm Mr. Balasubramanian Sharma, working as a research associate in the Department of Climate Change. For the past 4 years, I've been monitoring the phenology of woody species. Can I have the first slide, please? Yeah. Can you go into slideshow? Yeah. So I've been monitoring the community level phenological monitoring of woody species in Bannerghatta National Park. It's a tropical dry deciduous forest region. So the next slide, please. So I'll directly jump into the objectives and study area. I'm not going into the phenological aspects. So everybody knows of phenology. So it's the objectives are to understand the vegetative and reproductive patterns of woody species in Bannerghatta National Park, to understand the influence of weather parameters on the vegetative and reproductive phenophases, and to understand the interseasonal and annual variations existing in the phenophases. So our study area is Bannerghatta National Park. Pointer, please. Bannerghatta National Park. Pointers here. So our study area is the Bannerghatta National Park, which comprises of 260 square kilometers of area. It has a dry deciduous vegetation, and it is the confluence of Eastern Ghats and Western Ghats. So before going to the phenological aspects, I would like to mention that as our Director General Sir mentioned, we have established 3 permanent preservation plots, 2 at Bannerghatta National Park and 1 in Dorsanapalli Reserve Forest, which is in our campus. So these 2 plots at Bannerghatta National Park are the Thale Outdoors permanent preservation plot and Bugurkallu permanent preservation plot. Our idea is to monitor the phenological aspects which are representative of species in these particular plots. So what we have done is we have selected transects within a radius of 2.5 kilometers from the permanent preservation plots, tagged reproductively mature trees in transects that are representative of the permanent preservation plot. The permanent preservation plots are 1 hectare each, and whatever species that are there, we have represented in these transects. And we also marked the points on the transects for monthly soil sampling for moisture estimation. So just looking into the observations of phenology, phenophysis does not give you anything. So we have also installed an automatic weather monitoring in Bannerghatta National Park, that is at the Bugurkallu locality. So what are we monitoring? So we are monitoring the vegetative and the reproductive stages. In the vegetative stages, we are monitoring the leaves. In the reproductive, we are monitoring the flowers and fruits. So each of these stages are divided into five phases. For example, in the case of leaves, you're monitoring the leafless, leaf initiation, expanding leaves, mature leaves, and senescent leaves. So the total of these monitoring will be 100%. So we are monitoring the amount of the percent of canopy that is either initiating leaves or have mature leaves. Similarly, for all the other stages, the flowering and the fruiting. Apart from that, we're also looking into the herbivory. We are looking into the leaves that have been eaten by insects, the amount of leaves that have been eaten by insects, and also we are looking into the fruit eating nature of the fruit eating, that is frugivory. The unit of measurement is the intensity and sorry. I'll go back into the The unit of measurement is the intensity. So the intensity of phenophase is the average of the phenophase for the particular month into the number of species participating in that particular phenophase divided by total number of species. So this is a format in the field which we are using. We jot down the percent of the canopy that is either of each of these phenophases. So these are transects. We have the This is a transect which is around the permanent preservation plot. This is a permanent preservation, 1 hectare permanent preservation plot. So we have tagged around 254 individuals that are representative of the permanent preservation plot, and around 67 species are there among the 33 families. Until now we have observed around 48. We have done around 48 monthly observations and also around 43 soil sampling. Similarly, for the other transect, Bugurkallu locality, also we have tagged around 250 individuals, and which comprises of 68 species and among 30 families, and 48 monthly observations have been done. So we will go directly to the compilation of data and analysis. So for the soil moisture content, we have taken the average of the transects, Thale Woodhouse and Bugurkallu, and done the soil moisture estimation. As can be seen here, the soil moisture content is highest during the rainy season, during September and October. So here also, when you see the rainfall amount also, it is during September and October, the moisture content is the highest. So these are the weather parameters which are obtained from the automatic weather stations installed at Bugurkallu forest. So all the parameters, these 7 parameters are used for the analysis. So what do you want to do about the analysis? What do you want? What are the questions to be answered? So the questions we wanted to answer: what are the patterns of phenophases? The leaf initiation, the flower initiation, and fruit elongation. What is the influence of weather parameters on the phenophase? And is an individual weather parameter responsible for influencing the phenophase, or is it a combined effect of all the parameters? So when you look at the leafing patterns and the influencing weather parameters, See, these are the LF1. LF1 is the leafless stage, LF2 is the initiating leaves, LF3 is the elongating leaves, and LF5 is the dehiscent leaf. As can you see here, leafless stage is maximum during March. Similarly, leaf initiation is maximum during March and April. Leaf elongation is maximum during April, and leaf dehiscence starts. Yes, please. Starts during September, and it's maximum during January. So when you do a correlation, Spearman's correlation, rank correlation for the factors, the maximum temperature has a positive influence of leaf initiation, which is significant during the corresponding months. Similarly, sunshine also has a positive influence and during the corresponding months, whereas rainfall has a negative influence during the 2-month lag period. When you go into the flowering patterns and influencing weather parameters, similarly, you can see here the FL2 is the initiating flowers, FL3 is the opening flowers, FL4 is the mature flowers, FL5 is the dehiscent flowers. The flower initiation elongation is maximum during the April month. The flower maturation is maximum during May, and also the flower dehiscence is to the same direct during April. Here the values are different. So what are the factors influencing the flower initiation? We did again a rank correlation. The maximum temperature has a significant positive correlation or influence on the flower initiation during the corresponding month. Similarly, sunshine also has a positive influence on the flower initiation. Rainfall, the number of rain days, soil moisture, humidity have a negative influence on the flower initiation during 1 month and 2 month lag period. When you go into the fruiting patterns and the initiating weather parameters, fruit initiation and elongation are maximum during the month of May, whereas the fruit maturation is during September, continues till December. Fruit dehiscence is a continuous process. So maximum, I'll go directly to the fruiting fruit elongation, which is positively influenced by number of rainy days and minimum temperatures during the corresponding months. So as you have seen earlier during the in the patterns for the leaf initiation, flower initiation, we had seen that March was a month during the drier months. Both of the initiating leaves and initiating flowers occur together. So we wanted to do a correlation between these two, and we just found that leaf initiation occurs along with, or flower initiation occurs along with leaf initiation, and both are highly significant. And the correlation, the rank correlation was 0.92, which is very highly significant. And I would like to mention here. Next slide, next slide. The last slide, you can go for yeah. Yeah, that's fine. That's fine. Okay, slide. So we found the individual factors that were responsible for the influencing the phenophases. So we wanted to look into the combined effect of the phenophases. Previous slide, please. Yeah. So we wanted to look into the combined effect of the phenophases. So we did a principal component analysis of the weather parameters. Can you go? We did the principal component analysis of the weather parameters, and we found that 88% of the principal components were accounted for the variance. So we wanted to take only these 3 principal components for the analysis of the phenophases. So what we have done is, as can be seen here, this is the principal component loadings of the principal component 1, and only the moisture related factors, total rainfall, number of rainy days, average soil moisture, and humidity are responsible for the variation, which accounts for 52%. So, and other 3 are PC2 and PC2 and PC3. So what we have done next is we've taken the 3 PCs, PCS, that is the regression analysis of the principal component scores with the phenophase intensities, and did a multiple regression, and we found that the correlation, positive correlation. Sorry, I'm sorry. So we found that it is negative. The PC1 component is negative, whereas the PC2 component is, the coefficient is positive. Similarly, for the flowering phase flowering also, it is PC1 is negative, PC2 is positive, implying that the moisture related factors are important. So we finally confirmed, based on the 2 years data, confirmed that moisture related factors, the total rainfall, number of rain days, average soil moisture, humidity have a negative influence of leaf and flower initiation, whereas minimum temperatures and sunshine has a positive influence on leaf and flower initiation. So the way forward, we would like to continue these studies. So continue long-term community phenological monitoring in BMP to better understand the effect of changing climate on phenotypes, to collaborate with other researchers working on community-level phenology in different regions and vegetations, to understand the differences in patterns of, in order to understand the differences in patterns of phenotypes between communities, to understand species-specific variations in different geographic locations and vegetations, to create a network of researchers studying plant phenology for data sharing, collaborations, and publications, and also set up phenocams in BNP and other regions to complement studies undertaken via traditional observations in BNP and other regions, and also to create a network of Phenocam researchers for data sharing and publications. Thank you. Yes, please. Any questions?
So you have this only one place in BNP and
Oh, only in BNP. We have 2 transects in BNP. Only one place. Dry distillation.
just one place, one forest.
Yes, yes, only one forest. Yes.
Is basically monthly.
Monthly.
Have you also tried to see like what happens when you go back on this spontaneous phase change in the value that you get, let's say a date, and previous to that, how much time it has taken to accumulate to become a stimulus to change the frequency? It may not be monthly always, it can be 15 days,
Yeah,
or it can be 20
yes.
days, or 8 days. So
We are doing only the monthly observations. So we are not gone into the weekly or the daily or the weekly observations. So analysis has to be done. Yes.
I think those will be the learnings for second phase now, that we need to go down further.
So if there are no questions, thank you.
Good morning, all. Myself, Kiran Casey. I'm working as a senior research fellow in a DST project titled Strengthening the State Climate Change Center in the state of Karnataka, second phase. And this is a project period of 5 years from 2023 to 2028. And the objective of this phase 2 project is first one is vulnerability assessment and risk analysis, risk assessment of water, agriculture, forest, health, and urban heat island sectors at district level in Karnataka. And second objective is establishment and monitoring permanent preservation plots in natural forest for long-term research studies on climate change. And third is strengthening the climate change knowledge portal to make it more dynamic and interactive and user-friendly. And the fourth objective is knowledge dissemination through workshops and creation of an app-enabled database for climate-sensitive plant and animal groups through citizen science. In this workshop, we mainly focused on the objective, that is establishment of monitoring permanent preservation plots in a natural forest for long-term research studies in climate change. As Balasubramanian said, the phase 1 is continued in phase 2 project also. The same plots. What are the 3 already established plots in BNP and Durasan reservoirs. It is continued in phase 2 also. And apart from the monitoring the existing plots, the other objective is establishing the new preservation plots for a plant phenology based long-term study of climate change impact on forest vegetation response using Phenocam. In this objective, we are going to use the Phenocam for studying the phenology of different forest types in Karnataka. In that, we're going to use plant fate, that is a plant functional trait evolution, will be used to explore how adaptations at a different organization level and across multiple temporal scales, determining the different resilience of forest to climate change. In that, we're going to observe the short-term acclimation of plant physiology and also midterm changes in community composition and long-term evolution of plant functional traits. And apart from this, we're going to establish already existing, apart from the existing 3 PPPs, we're going to establish new 3 PPPs in a different forest type, that is dry deciduous, moist deciduous, and semi-evergreen forest, to examine the long-term effect of climate change. Apart from existing 3 plots, we are going to add 3 more plots for phenological studies using Phenocam. And we discussed about different organizations and also Karnataka Forest Department for selection of plots. Already Karnataka Forest Departments are maintaining the long-term linear increment plots in different parts of Karnataka in Western Ghats. And also we spoke with the French Institute of Pondicherry. They are also maintaining some preservation, permanent preservation plots. And also IWST, Indian Wood Science Technology, they're in Bangalore. They are also maintaining some plots in Kuduremukh and also in Sirsi regions. These are the plots. The rounded plots are maintained by circular mark that is shown by KFD and FI French Institute of Pondicherry. They are maintaining the Chamarajanagar, Kodagu, Dakshin Kannada, and Shimoga and Sirsi. In all Western Ghats, they are maintaining the permanent preserved plots, and also they doing phenological activities also, and also taking the readings of long-term studies of climate change using the biomass estimation and also the phenological aspects. They're doing. In this phase 2, through Phenocam, expected outcome for Phenocam in this study, there are main field observation based vegetation related data sets from the field of view of Phenocam. In that Phenocam, you're going to assess the leaf area index and net primary productivity and gross primary productivity, tree biodiversity, and tree traits, plant traits at species levels. We're going to study in this Phenocam. And also collection of weather data around the year from the automatic weather station established combined with the Phenocam, and also examine how forest phenology in changing over the specific forest types in Karnataka over last 2 decades by integrating the historical satellite data with Phenocam. We're combining the satellite data and also Phenocam data. We're going to study how phenological changes happen over a decade of periods in different forest types. Yes, thank you. In this workshop, we're going to study the main focus on the Phenocam. The Phenocam is a new topic, and it is we're going to understand how Phenocam technology will be used for study the phenology in different forest types, and also how to use it in monitoring the phenological changes in various ecosystems, and to understand the nature of Phenocam data and data flow and data storage, how it will be storage, and also data access and data sanitation and data processing. And also what are the parameters to be monitored in a national network. We're going to be part of that national network of Phenocam, and also the protocol at national level for monitoring. What are the protocols for national level in monitoring the Phenocam and phenological studies, and also interpretation the data with the national network, how they are doing and how we have to interpret with data with their and also the selection of permanent preservation plots for Phenocam installation, where the ideal places of we are going to establish the Phenocam to study the long-term phenological study. We're going to study this and also get information from this workshop. Thank you very much.
So thank you, Mr. Sharma and Mr. Kiran, for presenting what EMPRI has been involved in phenological study and what we intend to understand from this workshop. Now, I'll call upon my first speaker. That's Dr. C.P. Singh. Dr. Singh holds a MTech degree in remote sensing and PhD in environmental science. He joined the Space Applications Center, Ahmedabad, in year 2002 and is currently working as scientist engineer SF. He's played key roles in various recognized research projects related to satellite remote sensing application in the field of forestry, ecology, and environment. Under his guidance, a long-term ecological monitoring network known as the Himalayan Alpine Dynamics Research Institute was established, and is also spearheading the first of its kind Phenomet network in India. His field of interest also includes algorithm development to active forest fire detection, ecosystem modeling, paleobotany, and climate change impact studies. To his credit, he has more than 60 peer-reviewed journals, publications, and Dr. Singh has represented India in various international forums, including 39th Indian Scientific Expedition to Antarctica. He's represented several, he's a recipient of several prestigious awards from Indian Society of Remote Sensing and President's Appreciation Medal from Indian Society of Geoinformatics. Welcome, Dr. Chandraprakash Singh. We are honored to have you here.
Okay. Good morning, everyone, and happy Teachers' Day to all of you. Today, we are celebrating the birth anniversary of Dr. Sarvepalli Radhakrishnan, who was our former president. It's so auspicious day today to be here. I mean, we'll be learning from each other. In terms of field, you know a lot than me probably, so it's a learning for me as well. So thank you, Shruti, for giving an introduction. And I know a few people here who have worked with me, like Rajiv, Sureshi, and I'm very happy to meet EMPRI people, especially thanks to Honorable Jagmohan Sharma-ji, who has invited me for this particular interaction. So let us hope for that best, like whatever knowledge I have gathered in these years, I'll try to impart, and we'll try to make sense out of it, what exactly we can go ahead and do for this particular network of Phenocams. Can you have the slides? Okay, from here itself I can control. Yeah. So why I'm calling this particular thing as a pulse of the forest ecosystem? whenever we get sick or something, we go to doctor and it's try to see our pulse. But when we talk about forests, trying to know the health of the forest, how they are coping up with the current climatic situations. So what could be the pulse of it, So if you want to understand a priori that something is going to happen to this particular forest system, the phenology is the best tool to understand. Have you ever thought that how the plants decide to grow? Do they follow some kind of a calendar? They wake up in the morning and try to have tea or something like that, and then they think that, okay, today I have to grow a little more. Do they, see the mobile phone and see the time and then try to bloom? Gosh, aisa hota hai. But this is not the thing which happens. What they do is basically they respond to the various parameters, which we call weather parameters. And in long term, it is called climatic parameters. So it is important to understand what are the drivers for these particular growth parameters, which we see in terms of seedling, fruiting, flowering, leaf initiation, senescence. What drives them to happen? If you try to understand that, that becomes the study of science of phenology. And it is very important to understand these drivers will change from place to place. They become limiting in some other place, but may not be that important in other place. So it is very important to understand what is the interrelationship between these weather parameters and the plant's growth. It would have been great if the plant itself can tell the stories, how they have grown since beginning when the life started on this earth. But we have to look for it because they can't speak. So we have to look for signals. We have to use science technologies to understand what would have gone through in various time frames. They can't even put in the computer like what date happened, like we can have our own albums when we got married, when we had kids, when we educated ourselves. They can't put all these things, in the form of a computer data. But there are people, in the beginning, they were curious and they were trying to understand. And that has made the science of phenology very old, In 1940s itself, 30s itself, people were trying to keep recording Western worlds. Of course, in India also, but probably we were not very great in terms of recording. We were memorizing and putting in the form of poems, local poems. If you go to villages, you will have recitations of various poems, which will give you a clue of phenology. But there, they were more in Western world, they were keeping records of it through their municipal bodies and other things. There were artists like Leonardo da Vinci, who was, putting the various phases in terms of arts. There are various records, which are available through which European world could say that so much of shift in terms of flowering of sunflower or various other flowers have happened. That's how people become curious to understand why these changes, why these kind of shifts are happening. And that's how the climate change angle and other angle have come to the force. Have you ever thought why Champion State classified forest in 16 types in India? They're basically governed by the weather parameters, and the weather parameters are also governed by the local topography. Okay, so if you have higher altitudes, probably you'll have different temperature regimes, and so on and so forth. So all these influence the phenological patterns, the kind of forests, the species that are there in that particular place. It's all a culmination of what weather parameters are there in that place. So it becomes very important to understand what forest type you are investigating. So the local drivers will be different. So that's how I'm just trying to put things into perspective so that you understand that how important it is to understand the limiting factors in terms of weather for a particular forest type, a patch that you are trying to. So no single equation, no single hypothesis is going to explain phenology for forest. Coming to the definitions, this also gives you some kind of idea that how phenology has evolved and how we can define it and put it in our perspective. You know that phano in Greek means to show, to bright, to light, make, appear. So phenology is basically appearance of morphometric characteristics of tree species. So that is what we are trying to record. It is also understood as an area of science focus on the timing of the life phases of all organisms, because all are interlinked. So it's not only trees which are flowering or giving leaf initiation, it's also the interrelated species which also blooms and come to life or disappears. Phenology is the study of recurrent biological, plant as well as animal phenomena, life cycle stages in relation to climate and other abiotic factors. So in short, it's a horizontal science which transacts all other biological sciences, statistics, engineering, and everything else. So it's basically, in one word, if you want to understand one sentence, phenology is nature's calendar. And nature's calendar, if you can understand, you can predict. You can gain predictability in future climatic scenarios, what is going to happen. And why it is important. Even if you understood phenology, what gain that you are going to give to society? So this is the answer to that question. You can predict many things. Health is another important thing. We are all living in cities, but we are all governed. Our health is also governed by the forest around us, the trees around us. Have you heard of the volatile organic carbons? A lot of volatile organic carbons are emitted from the forest, from the vegetations, which are very beneficial to us. If you inhale, a lot of bronchial diseases can be cured. That's why the doctor says go to some natural areas and live for some days so that you can get cured. Okay, so they are very important elements. More than hundreds of VOCs are understood, isoprenes and so on and so forth. They are very beneficial to humans, for the health point of view. They stimulate your minds. They can cure a lot of diseases which are asthmatic or kind of stuff. But at the same time, some pollens are some people are not good in coping up with the pollens. So health is another important thing which you can if you understand phenology, you can give it to the society that what kind of scenarios you are going to have in terms of socio health point of views. Resource management is another thing for forestry, for the resource manager. It is important to understand what is the phenological status, whether our season growing length is increasing or decreasing, what kind of carbon sequestration you are going to have in the forest. It is also important for the conservation plan, agriculture, understanding hazards, and recreation, and so on and so forth. Hazards, why? Because in India, forest fire is primarily starts from ground, when there is a lot of accumulation of fuel, which is dry, so that any negligence can start as a trigger and you can have a havoc in the forest. So it is also linked to phenology. So if you understand phenology, you can predict that when is the likelihood of scenarios of forest fire due to negligence. So phenology is an indicator, and IPCC has also recognized phenology as one of the important process to track ecology of species in response to climate change. Because it is controlled by environmental and climatic factors, therefore considered as very sensitive biosphere indicators. So there are a lot of scenarios in terms of phenology shifts. What we are expecting out of phenological studies is whether they are shifting, the time of flowering, timing of migratory bird arrivals in response to it, timing of egg layings, timing of animal emergence. All this comes under the purview of phenological shifts, but we are trying to focus here more on the forest phenology, but though they are all linked to various other problems. So you can take up these as a, secondary problems associated with the phenology. So this is an example of how phenological mismatch can happen, and it has devastating, a catastrophic event may happen in the ecological sense. So if let's say a migratory bird arrives at a certain times, lay its egg at that particular position, and then there are hatchlings which are coming up. But at the same time, something happens, and the food available at that place in terms of worms or other insects, if they disappear, what will happen to the hatchling? So this is an ecological disaster which can happen in global warming situations where some studies have been done and people have predicted that there will be a lot of biodiversity loss if these phenological changes are going to happen. So why I'm calling this as a pulse of ecosystem is because of certain facts. And these facts are like, these are giving us the quantitative information on forest, which is vital for understanding the interaction between biosphere and climate. Leaf phenology alters the land surface boundary conditions by changing surface albedo, roughness, and surface water and energy flux. These are very important keywords, and they have very important scientific aspects. The moment the trees shed their leaves, you know, the roughness, suddenly the roughness of the surface of the earth changes so much that it will have a strong bearing on the kind of wind which is flowing on the surface will happen. Because these leaves becomes barriers, so Z0, which we call in terms of physics. So these Z0 values will be completely different. And you have different kind of wind velocity coming to the leeward side from the forest. Okay, so roughness is also important. At the same time, water and energy fluxes will completely change. These leaves are nothing but, you know, kind of, if you see in a mechanical sense, what exactly is a tree? It's a biotic pump. It's trying to draw a lot of water from the ground and trying to pump through the leaves. So this is going to change. The energy that is absorbed through photosynthesis suddenly gets changed. So if you try to see carbon dioxide in the atmosphere, suddenly when then leaf senescence is there, suddenly you will find that CO2 concentration in the atmosphere is high because there is nothing which is sinking it. So that's why water and energy flux is very important. If you understand phenology, you can predict what is going to happen in terms of gaseous exchange, which is happening to the biosphere. At the same time, albedo. It will completely different surface that you will have from the green to the brown, the albedo will differ. So whatever sunlight is coming is going to come back more or less will depend on the kind of surface that you have there. Leaf onset and offset marks the bounds of the growing season, and therefore annual carbon uptake is going to change, as I said. Increase in the mean annual temperature of just 1 degree Celsius can lengthen the growing season by 5 to 10 days, according to some studies. So these are very important elements. What is going to happen in our biosphere? The timing of leaf offset in extratropical trees is governed not only by the temperature, but also by the length of the day. So how much sunlight has been accumulated over the days are also important and important for the growth of those plants. Phenological variables have the potential to characterize many ecosystem variables, not only the water energy fluxes and albedo, but also on the net primary productivity, fuel load, as I said earlier, habitat properties completely gets changed. And animals try to migrate because of the responses from phenology. And these variables are also going to change the functional traits of a particular patch of the forest. Phenological parameters are also important for the ecosystem modeling and earth system processes. Nowadays, there are a lot of models which are built to understand the weather parameters. You must have heard about the WRF. Okay, weather forecasting models are there, which meteorologists basically works. But they are sometimes don't get answers. They become clueless that why such scenarios have happened. They are not able to completely understand that. So answer sometimes lies in these earth system processes. So nowadays, ecology is a big input for the land surface modeling, where they're trying to understand the dynamics of these forest ecosystems and trying to put them in the code so that they can predict it better, in a better way. So it's a feedback mechanism. You have to understand that, and then only you can model the land surface processes very well. So there are a lot of gap areas when we try to understand for the phenology in India. The biggest gap area that we have is there is no well-calibrated and validated product on phenological matrices. There are very few people who know actually that there are phenological products available from satellite itself, but they are just mathematical products. They are not validated. The model itself was not calibrated, but they just put it something for the academic people to understand what does it make any sense or not. So we have to make our own product, first of all, which should be calibrated and validated enough so that it becomes an essential ecological variable. We have to qualify it to make it, call it essential climate variable or ecological variable. And there is no systematic study of impact of change on phenology and its feedback to Earth systems. So this is the biggest gap area. So science questions will come from these domains itself. And there are some science questions like, what are the key meteorological parameters or predictors that control the phenological matrices? Sharmaji just showed that some modeling that he has tried to understand through PCA analysis and all that, but that is not enough. We have to understand very deeply how these biological systems are functioning. They're not responding in the, you can say, in a constrained way, the calendar that we understand, that something in monthly, so it's not monthly. You have to understand that it is, you have to basically do a lot of, build a lot of models, which will actually tell you that it's not monthly, it's weekly or 10 days or 7.5 days. If you have high resolution data, because through these phenometric networks, we are going to get 30 minutes data. So that is going to be very important for you to understand that what is that minute level changes which are triggering something, to change its phase. Can we make predictive models for phenology? That is what is the ultimate goal, should be ultimate goal of these kind of studies, that whether we are able to gain some kind of predictability, that given in climate change scenarios, in 2047, if 2 degree or 2.5 degree change in temperature is going to happen, can I say through my model that, look, length of the season is going to be wider, we'll be sequestering more carbon, or there will be early shift in the entire season. I'll discuss later on all these things, what all we can expect from these studies. They're going to give us beautiful insight in terms of ecology of the forest, and it will give us give us predictability and preparedness. It will prepare us to cope up with these kind of changes in a better way. So ultimately, whether phenological cycle is shrinking or widening or simply shifting left and right, what would be the likely future projections of land surface phenology in various climate change scenarios? Can we model the net primary productivity using these phenological models? Because there are a lot of modeling abilities that people have gained for net primary productivity, gross primary productivity, but whether using phenological cycle itself, can we model the net primary productivity is the key question. So there are various methods. As you know that we have been observing on the ground since long. We have ground-based cameras as well. We have Phenocams, we have UAVs, we have satellites, but all have their own limitations and positive points as well. So the best thing which has come to us these days is the Phenocam, because unattended, you can put it at the vantage point, which will take stock of the entire landscape, because other are having its own merits and demerits. Though satellite is also from a long time, it has its own challenges of temporal resolution, because earlier they were, observing 22 days, every 22 days, same place will be, imaged, which is not enough for a phenology scientist, right? So Phenocam gives us the leverage of taking as many pictures as we can store and process. We have tried to understand, actually, recently, when we get into this kind of subject, we thought, let us try to understand what have been done in the past in India. And we were surprised to understand that very minimum level of study of the phenology has been done. I have not put it for the agriculture. Agriculture, a lot of activities have been done. But for forestry, hardly 50 work have been published and cited, which is there in India, which includes field-based measurements. And generally, it is for the PhD purpose. So people do it for 3 years, 4 years, 5 years, and then after that, there is no record of it. So such studies are there, but still it is very, shocking to me that only for such a big country where we have such an academic culture, only 50-55 studies have been done. So we have to increase it so that our observations get better and better. There is an interesting case I would like to project here. We were working for Kanha National Park in Madhya Pradesh, and this is one of the systematically managed forest area, Kanha National Park. And there, to my surprise, there are a lot of beads which are having good records of phenology. Somebody thought about it, and they have made registers to note down. Every bead guard is supposed to come back and note it that today he found so and so plant was senescing, or so much leaf initiation was there, fruiting of flowering was happening or not. And very interesting records. With a lot of pain, we could collect the data, and we tried to make sense out of it. But still, I found that there are a lot of human bias into this, because once the bead guard is transferred, the next bead guard, who is supposed to be not very well educated, but he is a good observer, but his observation skill is also limited by his own skills, of observation. So we found that a lot of human bias, but still this tells a story of phenology for a long time. Though it cannot be published because it will be challenged that what kind of bias, human bias has gone into it. But it's very important. So what we did, we equipped those bead guards with a camera. We gave them a simple Sony camera, and we put a pillar there that you have to stand here, face northward, click a picture. That's it. That's the simplest thing can be done. Phenocam installation will take its own permission and money and other things, but just giving him a 5,000 rupees camera can do these kind of activities. And we did it for 5 years. And that also gave us a lot of understanding. We used this system in at least 6 national parks, where we gave to bead guards these cameras. But there was a challenge, because humans taking picture, someday he will take like this, someday he will take like this. Even half a degree of movement in X or Y or Z direction is going to change the entire, though the plant will be same, tree will be same, registering them and then analyzing, making sense out of it. Manually you can do, but manually taking so much of data is painstaking. So what we did, we use our geometrics techniques, taking these branches as a key, you can say registration point, fiducial points. We register them automatically. That's how you are seeing that some pictures are having blackish boundaries. And we register them, we take a region of interest and try to make sense out of it in terms of greenness, how it is changing and not changing. But that time we were not having meteorological stations there. So you have to rely on the IMD kind of data, which is at 1 degree level. So meteorological bearing on these kind of changes were not sufficient for us to understand the science behind it, but we could observe and study a lot of things. And we gave them the protocol, how to do the photography. I'm not sure how much they could follow, but at least we were fully equipped and prepared that they have to choose the plant, what way, what kind of direction has to be there, and how they have to keep even the record, handy in terms of, registry. Actually, this is the best kind of photography, which is for the phenology, which can be done. This is called fisheye lens. Those who are into ecology and understanding the leaf area index, they use this kind of photography. If something like this can be set up in the forest permanently, this is the best equipment, better than I consider the Phenocams. Because Phenocams is going to give you the fused canopy of many layers. If you are watching from this vantage point, there are many layers of trees which are there. So there, a lot of times when there is an obscursion of, or you can say that even the leafless plants are there in between the 3 different layers, you are going to miss them. But this is going to give you actual view of what is the leaf area of that particular time. But this is automatizing, putting them on the ground without getting disturbed by animals and humans. It's a challenge. So I have never seen that such kind of concepts are being practiced world over, but I feel that this is the best thing to do. Coming to spaceborne data, availability of time series data has been there since long. Since 1980s, even the high resolution temporal resolution data is also available. And then phenological matrices can be retrieved. Challenges are there in terms of filtering, smoothing. There are a lot of spoil sports for remote sensing, including clouds. which will optical reason, it will be susceptible to the presence of cloud, and you will get the NDVI that you rely more will be completely different. So you have to understand, learn the tricks of the trade, how to, normalize those things and get a better signal. So that's why Phenocam becomes very important. And this is the kind of data which I was talking about available from Modis. These are examples of those kind of data sets. This is, I've tried to analyze and seen that a lot of errors in these data sets. This is from the NOAA EMPRI. And we have tried to make phenological calendars, but again, reliability is questionable. And this is the kind of reliability that we come across. If you see the Western Ghats, it's not very reliable, even the Northeast area. So these are the satellite data sets which are available for you to look into and do some kind of landscape level phenological analysis. NOAA EMPRI 9282 onward, which is having almost daily data. Recently, we have launched, ISRO has launched OCM3. So we have continuity, and then 2010 onward, it's a beautiful data because 350, 60 meter kind of data is available and daily data, which is going to give you vegetation indices. Resource set to AVIPS. AVIPS is fantastic. Every 5 days you can get data. Sentinel-2A and B put together, and they are giving data in open domain. Those who are well versed with the Google Earth Engine, they can just simply, write few lines of code and data is in your desktop. Okay. So this is every 5 days and fantastic data, 30 meter. So spatial resolution is 30 meter, fantastic data. You can do a lot of phenological activities. Earlier, we were using Spot VGT and ProbaV, which were available from 1998 onward. Modis is well known, but only thing is coastal resolution, 1 kilometer kind of pixel resolution, but daily data available, still available. We have utilized INSET-3A CCD and INSET-3D. INSET-3D is a meteorological satellite from India, which is also giving you vegetation indices, and every 15 minutes. So it's like Phenocam sitting on the geostationary orbit. Problem is 4 kilometer pixel resolution. So you will not be able to appreciate, but if you want to see globally how things are changing, probably this kind of satellite data also help us. So these are the kind of matrices that you can generate, whether it be it the ground photographs, be it Phenocams, be it satellite data. This is the kind of model fitting you can do. You can see a typical Gaussian bell-shaped curve, but in reality, when you work with the data, you will not get exactly this kind of bell shape. So if you see a line behind it, in a shaded color, that is the actual line. So you have to try to make a fit of it, try to understand from the polynomial fits that what is the second derivative on my left side and right side of the curve, and making sense out of it. What you call start of the season. That itself is a challenge to the ecology community, that when I call it a start of the season, when I call end of the season. Okay, some people say 10% of the leaf remaining as a green and 90% has become brown, I'll call it senesced period. Some people say no, 20%. Some will say 15%. So how do you define it? It's a problem. So you have to have a consensus. So that's why these kind of networks are very important, where we build a consensus. What is that we will call start of the season, so that everybody is on one page and we start making sense out of it, so that our data becomes comparable, our inferences becomes comparable, right? And host of statistical you can say parameters can be derived out of this curve, which are having some ecological significance. Just for the sake of derivation, it should not be done. It should have some ecological inference, then what we should do. Like if I say large integral and small integral on this particular curve, whether large integral is going to tell me the carbon sequestration or small integral. So that kind of questions we have to answer, and we have to derive those kind of parameters. So there's a long processing chain, and there are a lot of literature available how we should be processing. Some people will take 2K method of smoothing. Some people will take Savitsky-Golay method of smoothing. Some will take some other method. There are a lot of statistical methods. So we have to also think that which data, which method
is the best method for having the actual representation of what we see on the ground, right? So that's why Phenocam are important. Then because we work with satellite data, we do some fitting, which is not actually matching with the ground. And there are a lot of challenges which are when we are working with these Phenocams, we are gaining and getting it on the ground. And there are a lot of challenges which were available as a challenge are now getting resolved because of the availability of Phenocams. So that is also happening. So this is an example of satellite view. You can see how greening and browning is happening. Phenocam view of Gir forest area, which is under this network, and field photographs, how they look like. So you can have all these kind of observations, and which will basically ultimate aim is to utilize satellite data because that is available from the long term. Now, if you decide for the Karnataka that I want to go back in 1998, you don't have any other way to go back. You have only satellites to be available. So you have to ultimately come out with satellite kind of data observations and come out with the phenometrices. Phenocams are going to give us a lot of understanding on the ground, as well as they are supposed to be working as a calibration and validation for these kind of models, which we are bringing from the satellites. So these are the kind of changes we can expect on the curve. If you see whether it can shift in terms of climate change in response, or it can just shrink, or it can just widen. Okay, so these are the kind of things we can expect. These are some of the case studies as I would just quickly give you. We have tried to do it from the INSET 3A CCD. Then, yes, this was a very interesting thing. Generally, we are well versed with the optical data, which gives us 0.4 micrometer to 0.7 micrometer. In between, our eyes can also, get the same kind of information. Only thing we add is an IR band. So which gives us, because of the cellular structure of the mesophyll structure of the leaves, it gives a lot of brightness, whereas red band is absorbed in the photosynthetic process. So this contrasting thing gives us a vegetation index, which gives us the vigor of the forest. So that vigor sometimes gets subdued because of the cloud presence. So there are areas like in Karnataka, it will be a lot of time cloud will be there because it's a coastal region. You will have a lot of persistent clouds. So your NDVI will be suppressed. You will not get the actual greenness. So what happens sometimes, this kind of data set becomes very important, which is microwave data. And this was a very interesting data with that time available was QuickScat KU band data, which is not having a great wavelength. It is having a wavelength enough for even the leaf bud burst to interact with. So it becomes a roughness for it, and you will get the backscatter coefficient. So we tried to utilize, and we found that this is a better predictor than the NDVI. So we need to also demand such kind of sensors which are good for phenology to the space agencies of our country. So but for that we have to do a lot of work and prove it that what kind of resolution we want in terms of spatial resolution and temporal resolution. This is an example from the 3A CCD, where we could actually make a good output, having known some of the literature available data sets on the ground phenology. We calibrated, and this is the calibrated and validated model. But this is not enough just by having 4 or 5 points in the country. So we want to have a network. This is an interesting study we did for the Uttar Kannada. I especially wanted to show because you are also showing Uttar Kannada district. It's a very special case because ridge is cutting the entire Uttar Kannada into 2 parts. So you can see the left part, which is hashed one, is towards coastal area, which is having evergreen forest, and the other part is having trideciduous kind of thing. So it's a very interesting place because at the same district you can get both kind of forests, evergreen as well as deciduous forests. So laying the plot, having similar kind of climatic region, you are having 2 responses. And this can be a best case for doing it. So Bhatt et al. Has published some paper. We utilized those numbers because it was for a 3-year study. And we trained our phenological model from the satellites, and we got good results. It was published in 10 Science. This is an example of how in response to phenology, NPP can change. And then you can see NPP change in the country also is look like a kind of, phenology itself. This is the example I was just trying to show you that from Siamachi, we observed carbon dioxide concentration in the upper atmosphere, and we found that this is very much controlled by the senescence and leaf initiation itself. So you can see, though the CO2 concentration ppm is increasing in the curve, it looked like a monolava curve itself, Keeling curve, it is famously known as. It is for India. Okay, and you can see this is also having a Gaussian fitting, which is actually indicator of the forest, as a sink, how they much, the CO2 they are getting absorbed and how much it is getting liberated. So phenology is also as important as I was telling. We have actually made a model, which is a prototype model, which was tested on Madhya Pradesh, where we put a dynamic component of phenology, and that gave better results than without putting phenology. Okay, so dynamic components in any forest fire risk modeling could be weather. Generally, everybody takes weather, but phenology hardly anybody talk about. So we put it phenology also as one of the predictor, and that gave a better results. There are a lot of WebGIS which is available these days, even from the International Forum, but we have from Space Application Center, there is a portal known as Vedas. So if you just search on Google Vedas, SAC, you will get a portal. There you can monitor for any area, for your any area of interest. You can simply from SPOT, from AVIPS, so Indian and foreign satellites, all NDVI VIs are available. Just click on that particular pixel. It will give you long-term pattern of how it has grown. So you will know the seasonality. Just by knowing these curves, you can even predict that this is agriculture area. So if somebody has encroached in your forest and doing agriculture, you can prove it in the code of law by this technique. Because forest will have one season, whereas agriculture may have 2 or 3, because he's taking 2 or 3 crops. So just by putting it, you can know. So this kind of forensics of ecology or forensics of environment you can do, right? These are some papers which we published initially for the tree line, because phenology is also very sensitive, and to understand the sensitivity, if you go to the sensitive ecosystem itself, that will give you much more meaningful data. So what we did, we used alpine tree line ecotones, which are the fuzzy line between the grassland patches and the tree line itself. So where 2-meter tree height trees are growing, they also have their own phenology, whether it is betlite place, which is deciduous in nature, AB spin row, then you have rhododendrons, they have their own interesting phenologies. So we mapped first the alpine tree line ecotone, and then on top of it, we tried to extract what is the phenological variables. And we found that, yes, there's a lengthening in the season which is happening. And this is happening across the alpine ecosystem. We published records for Arunachal Pradesh, Sikkim, Uttarakhand, everything. Now coming to the predictive ability. We have done some case studies where we have tried to gain the predictive ability for forecasting what will happen to the forest phenology if X amount of temperature, X amount of rainfall is going to change in future in IPCC scenarios. So in that case, what we did, we try to understand, first of all, how the biology works. Okay, so if you see a tree, the phenology of it is not just governed by the instantaneous temperature. It's not a mechanical device that today you increase the temperature of your place and suddenly it will senesce its leaf. Or suddenly, if I pour a lot of water, it will start having leaves, new leaves. So it doesn't work like that. It's not a mechanical system. It's a biological system. It is governed by a lot of hormones, a lot of nutrient availability on the ground, which is governed by the microbial activities. So it's an ecosystem response. It's not the response of a particular one single tree species, right? So a lot of papers have been published in Nature and Science where people have proved that before the rainfall happens, they somehow sense it. And there's a lot of microbial activity which start happening on the ground, and they start releasing nutrients, macronutrients, and plant will start investing in new leaves. The investment may fail if rain fails, but they try to take that risk. They'll put a lot of investment in new leaves in response to what is going to come in coming 5 days. That is the rainfall or whatever stimulus it can be. So it's very important to understand what is that stimulus in my ecosystem and what is the response, delay time or accumulation of that particular stimulus, what is that particular time available. So that's what we try to understand through, you can say, moving window kind of in time domain data collection. So if I taking temperature, I'm accumulating the mean, minimum, maximum from previous 1 day, 2 day, 3 day, 4 day, going up to 8, and then plotting my R square. When I get a asymptote, I know that this is the point where maximum significance of my data is there. And then I click on that and freeze that window, that this is the best response that I'm getting. So this kind of activities has to be done. So this is the seasonality of NDVI, simple NDVI. This is the phenological calendar maps over India. What is left is the validation. So when we do this kind of modelings, you don't get the actual dates. So what you get is Julian dates. So only 1, 2, 3, 65, right? So these dates need to be interpreted for these kind of calendars has to be there, which is called Julian day, a perpetual Julian day calendars. Only for February you have to take care. And then you can actually arrive at the date which you understand as a calendar. These are some of the studies that we have done. I already spoke about it for Arunachal Pradesh. This is how NPP from Phenology can be governed. The area under the curve can be indicative of net primary productivity, which is like carbon sequestration. And we try to actually quantify that using these kind of models. And we prepared net primary productivity map of entire India and calculated the actual values, which was like 490.5 gram carbon per meter square. So in conclusion, phenology governs biosphere processes and climate change impact scenarios need to be understood. There is a need to have systematic ground-based observations for calibration of phenological models and validation of results. There is a strong need to gain predictability. Phenocam data of AWS and with AWS can help understand drivers of phenology. The point of measurements, although landscape level bigger than the field shot, using Phenocam would help in generating a lot of thresholds for leaf initiation and senescence. As I said, we need to have a consensus on what is the definition of initiation and senescence. Thank you very much. So if any questions, please. Thank you. So maybe we'll discuss later if you're not asking questions. Yeah, there are questions?
I see that there's a lot of model fittings that has to be done in the satellite and then to see on the ground of the Phenocam. So how does the chances of overfitting the data, what are the models you actually use to fit the data?
Yeah, very important question. In fact, many times we feel that whatever the model fitting results are there, first of all, we are not sure that whether it is correct or not, because we don't have any data. We don't have any long-term observations, which is done at the very high resolution that every day one picture is available or data is available. Even on the ground where whatever, like I was quoting about the Uttar Kannada work in Bhatt et al. Paper, it's not every day that the researcher is sitting in the ground and noting the phenophases. So sometimes what happens when these actual suddenly, senescence will start happening, and within 1 day or 2 days, you will get the signals, and you are coming after 10 days or 5 days or 7 days. So you miss that event, actual event when it happened. And that is very crucial. So very important question. Right now, that's why we are putting this much investment and trying to make a Phenomet network so that our model fitting, whatever we are doing, can be adjusted according to the actual situations which is there on the ground. So we are in that process, and that's why this network.
Any other questions? Thank you so much for presenting. I think we'll break for tea and then. So those who people are online, we are breaking for about 10 minutes and we'll rejoin. Okay.